Auzubillahiminash-shaytanir-rajim. Bismillahir-rahmanir-rahim. Dear audience, Assalamu alaikum. This is 4th International Conference on Applied Zoology, a good platform to present the research work. I am Muhammad Ramzan, a student from University of Education, and the topic of presentation is antibacterial activity of Citrophola citricula silk, which was actually a spider. This study was carried out under the supervision of Dr. ICAZ-2021, Assistant Professor, Zoology, University of Education. I must acknowledge her efforts to carry this study. I am also thankful to my teachers, Professor Dr. Muhammad Arshad, Pro Vice Chancellor, University of Education, and Professor Hafiz Muhammad Tahir from Government College, University Lahore, for their brilliant ideas for this study. And I also acknowledge the efforts of my research fellow, Mr. Umar Daraz. Actually, bacterial resistance against antibiotics is a very common problem all over the world. There is an urgent need to discover some new antibacterial compounds with better efficacy. Antibacterial agents obtained from natural resources always prove best than synthetic ones. And many researchers have reported that some species of spiders have silk with antibacterial activity. So with this respect, the silk of spider Citrophola citricula was analyzed for its antibacterial potential. And the purpose of this study was to discover some new antibacterial compounds and drugs from natural resources, because such drugs are less likely to be resisted by the microbes. The objectives of this study were collection and rearing of Citrophola citricula spiders, and then silk harvesting in lab, and then analysis of antibacterial activity of this silk. The spiders were collected from Botanical Garden, University of the Punjab, then Changamanga Forest and Yellow Park, Lahore, and from some other areas. The key feature to identify the spiders Citrophola citricula are some prominent processes present on the dorsal posterior surface of the spider. You can observe in this diagram, and also you can observe in this diagram, and also the small spiny legs of the spider. And because these are odd weaver spider, the web is also an indicator. We reared these spiders in wooden boxes. You can see here these wooden boxes, and silk was harvested and collected with sterile glass rods. Then this silk was dissolved in 10% sodium hydroxide solution to prepare the solution of silk. This solvent was observed that this solvent is very good to dissolve the silk, and we prepare different percentages of this silk in this solvent by serial dilution. The test unit, or the microbes against which antibacterial activity was analyzed, were 4 in number. First one was Staphylococcus aureus, then Escherichia coli, then Pseudomonas aeruginosa, and Klebsiella pneumoniae. Disk diffusion method was used to evaluate the result in terms of zones of inhibitions on nutrient agar plates, and bacteriostatic activity was actually analyzed for this silk. The nutrient agar media, the composition of this media was 0.3 gram yeast extract, 0.5 gram peptone, and 2 gram agar was mixed in 100 ml of distilled water to prepare solution. And this solution was then autoclaved and was poured in the petri plates in a laminar airflow, then incubated at 37 degrees centigrade for 24 hours for screening. After which only the non-contaminated plates were further processed for the experiment, and these plates were inoculated with 100 microliter of bacteria from the broth media, and then separating was carried out. After that, each plate was divided into 3 distinct regions, or 3 distinct parts, and filter paper disc with 6 millimeter diameter was placed in the center of each part. So one plate get 3 filter paper discs. One disc was manipulated with positive control, the second one with negative control, and the third one with treatment. So these three groups was applied on the same plate to be analyzed in a very similar environment. Positive control includes 1.5% amoxicillin solution, and negative control includes the sodium hydroxide solution, which was actually the solvent in which the silk was dissolved, to check the antibacterial activity of this solvent itself, if it has any. The results were analyzed and manipulated in terms of zones of inhibition, and in statistical analysis, we applied one-way ANOVA followed by two-case test with 95% confidence. These are the results. When we prepared 1.5% solution of this silk in sodium hydroxide, we observed that this solution only inhibits the growth of Escherichia coli, but the other three bacteria, Pseudomonas aeruginosa, Staphylococcus aureus, and Klebsiella pneumoniae, these three were remained unaffected by this solution. You can see here in this diagram the zones of inhibition for Escherichia coli, the zones of inhibition for the treatment against Escherichia coli. These zones are statistically and significantly enlarged as compared to the negative control, which means that negative control also has antibacterial effect against Escherichia coli, but the effect in the treatment is not only due to this solvent, but also due to the silk in which the silk which was dissolved in this solvent. And this zone of inhibition is also comparable with the positive control. So here we can say that this silk has antibacterial activity against Escherichia coli. This was not the result for Staphylococcus aureus. Here the zone is not statistically enlarged as compared to the negative control. This was also the result of Pseudomonas aeruginosa and Klebsiella pneumoniae. You can also see these results in this statistical table. The superscripts for negative control and treatment in terms of Escherichia coli are different. These superscripts are different. But for Pseudomonas aeruginosa, superscripts are same. Pseudomonas Staphylococcus aureus, superscripts are same, and Klebsiella pneumoniae superscripts are same. These superscripts are applied according to Tukey's test with 95% confidence. When we applied 1% silk solution, we observed that this solution inhibits the growth of Staphylococcus aureus only, and the other three bacteria, Pseudomonas aeruginosa, Escherichia coli, and Klebsiella pneumoniae, these three bacteria were remained uninfected. You can observe in these figures for Staphylococcus aureus, the zone of inhibition is statistically and significantly enlarged. The treatment zone is enlarged as compared to the negative control. So this effect is due to the silk besides the solvent. And this treatment zone is also comparable with the positive control. This is not the result for Escherichia coli. Zone is not statistically enlarged as compared to the treatment, and this is the same result for Klebsiella pneumoniae and the same result for Pseudomonas aeruginosa. You can also see the described result in the statistical table in terms of superscripts over the means. For Pseudomonas, for Staphylococcus aureus, the superscripts are different for negative control and treatment, and the zone of inhibition for the positive control is always the most large. But for Escherichia coli, Pseudomonas aeruginosa, and Klebsiella pneumoniae, the superscripts for negative control and treatments are same. You can see here the superscripts are same. And when we applied 0.5% silk solution against the same bacteria, we observed that this solution inhibits the growth of all the four bacteria, Pseudomonas aeruginosa, Klebsiella pneumoniae, Escherichia coli, and Staphylococcus aureus. You can see the zones of inhibition for Escherichia coli statistically enlarged as compared to the negative control, and the zone of treatment is comparable with the positive control. For Pseudomonas aeruginosa, the zone is significantly enlarged as compared to the negative control, and the zone of treatment is comparable with the positive control. This is the same result for Staphylococcus aureus and same result for Pseudomonas aeruginosa. You can also observe the superscripts in this statistical table. The superscript for all the four bacteria in terms of negative control and treatment are different. You can see different superscript here, different superscript. So collectively we can say that the silk from Citrifolia citricola spiders has antibacterial activity against the described four bacteria when the silk was dissolved in 10% sodium hydroxide. Because the solvent must have the capability to dissolve the silk. If that silk is properly dissolved, then this show its antibacterial activity. We also recommend that a lot of work is needed to evaluate the antibacterial nature of silk from other species of spiders also which are still untouched, and the identification and suppression of active ingredient in the silk. It needs also much work, and the production of these biofriendly antibiotics at commercial levels may be possible through genetic engineering and some other disciplines. So that's all. Thank you so much for your attention.